So I'd like to invite Professor Biswas to the stage.
I'm really grateful to the organizers for giving me this opportunity. Today, I will discuss on a slightly different, in a sense, for me it is a little, newer area, which is basically understanding flow dynamics, viability and metastatic potency of cancer cell lines using microfluidics. Here I will this talk has 3 parts. In the first part, I will try to show how cancer cells propagate through or migrate through from organ to organ through microcapillaries. Second part is how the epithelial cancer cells get transformed into EMT cells, and final part is something related to drug delivery. Before I start, let me give you some dimensions. Usually the cancer cell lines, there are different type of cancers, as you know. Here we will try to focus on 2 type of cancer cells, primarily breast cancer cells and cervical cancer cells. Now this work primarily started with with the intention to do something related to breast cancer cells. The reason is, even if you go back by 15 years, usually, persons suffering from breast cancers, the surgery used to be the only medicine or only the, way of recovery. And people used to think after surgery everything is safe. But things started, appearing differently, and it was understood that even after surgery, specifically for breast cancer cells, there is no other route to reach the lungs if they do not cross some bunches of pulmonary capillaries. And diameter of these capillaries are in the range of 5 to 7 micron, whereas the diameter of the breast cancer cells, that diameters range from 26 to 32 micron. Now something is having diameter say 30 micron and passing through a capillary which has a diameter of 7 micron, it is unbelievable. And that is how the usual belief used to be that surgery is the sure shot medicine for breast cancers. For other cancers, why I mentioned about that, there are multiple conduits through which the migration can take place, but there also this mechanism is valid. So when we did this experiment, as you know, in our country, the cancer cells are available from National Center for Cell Sciences. And you have to submit your requisition to get the cancer cells. When we submitted our requisition to get the cancer cells, they could not supply us. There was a huge scarcity. Even now, getting specified cancer cells are always difficult. So we could get some HeLa cells. So first part of the study is with HeLa cell. We will come to breast cancer cell in the second part, where we will show EMT transition. So, can I take this water? So, as I said that in this first part, we will show basically these underlying themes: entry time, transit velocity, elongation index, etc. of cancer cells. So, this is the schematic. You can see that this part, our whole focus will be on this part. This entire length is as you can see 3 millimeter, and this part is 200 micrometer, and the diameter is 7 micron. It's very difficult to fabricate this. Today in our country, very few places this can be fabricated. IIT Guwahati now has full-fledged facility, but when I started this work in IIT Guwahati, we did not have this facility in place. It was done in ISC. Professor Prosenjit Sen, I am indebted to him, helped me a lot to get this. And you can see these are basically photolithographic print of these channels on SU8 master. It can be done on silicon master also. Then this with cross linkers PDMS is deposited. Then this PDMS pieces, you can see through which these channels pass. They are separated out, and you can see here this is the inlet hole, this is the outlet hole, and through this the microchannel is there, which is having diameter of 7 micron. Naturally, you cannot see with natural eyes. So, and this is at the back of the channel, and to get the full photography, we had to mount it on a glass slide, and that was done in oxygen plasma chamber. So, this is the basically test facility. Between these two ports there is a channel, and on this channel is covered by this glass slide, and all the photographic studies are done through that. So, here the HeLa cell, the background of HeLa cell has been given. It's named after Henrietta Lacks, and cells were available from National Center for Cell Sciences. I mentioned it about, and the cell culture is a very specific technique, and it is done using DMEM medium, which is high glucose containing 10% FBS, fetal bovine serum, and 1% streptomycin or penicillin in humidate air containing 5% carbon dioxide at 37 degrees centigrade. So, this is the test facility, as you can see, and it is mounted now under the microscope, and this is the inlet port and this is the outlet port. DMEM is the medium which is carrying the cancer cells, and these are injected through the syringe pump. So, I will show you exactly how cancer cells migrate. This is a single HeLa cell. As you can see, it's about 30 micron diameter, and it is coming, and okay. So, here I will show a triplet, 3 cancer cells, 3 HeLa cells together, and you can also see that the first cell passes through, but the second and third cell, while passing, they are almost fragmented, and there is a chance that these fragmented cells are dead cells because nucleus gets affected. Here this is a real cluster which is called CTC, a cluster of cells and cancer cells, and they are passing through from one side to another side. This is 200 micron length, and while passing through, several cells are fragmented. That means this distortion affects the nucleus, and with dead nucleus, of course, cells do not have any capacity to lead the cell growth in the later part, which is beneficial, but definitely there will be some live cells. And this study, as I said, even though this expansion, some cells die, but we have to we studied basically viability. So now this viability study was done through a fluorescence based study. Basically cells were stained, and these were stained with acridine orange and ethidium bromide, and you can see on the left hand side it's a mixture of cells, green, red, orange, all colors are there, but primarily green. These are all live cells. This is B and C is the transit zone, and D is the zone where again we get all the cells back, but here you can see basically some live cells, some apoptotic cells which are basically dead cells, and some necrotic cells which are dying cells about to die. And then further staining experiment was done, because just to cross check with trypan blue, and trypan blue staining is done in a very specific way, because it stains the dead cells and live cells exclude the dye. That is why it is called dye exclusion assay. And then the dead cells are counted, and obviously you get the statistics, how many cells are dead and how many cells are alive. And as you can see, this is the entry region. It's a sort of convergent divergent channel, and at the middle you will see acceleration, and again at the exit, which is like a diffuser, here again you will see the deceleration. We can, deformation index is basically while passing through the passage, the deformation is in one direction, its length is increasing. So deformation index is given by L by D, and we used a collection of cells. So basically you can see with cell size, the elongation index varies. Then entry time was also calculated, and you can see here also, although the relationship is slightly nonlinear, but with cell size the entry time increases. And this side we can see calculation of transit velocity, transit time through the 200 micron space. And here also you can see smaller cells pass very fast, and obviously bigger cell take larger time to pass through. Now after collecting these cells from the exit, another assay is used, which is called basically where we can see whether again the cell growth activities are observed. And here after 12 hours, 48 hours and 72 hours, we could see the footprint of the cell division, which is the production of MMP2. This is an enzyme which is capable of degrading the type 4 collagen that is abundantly present in the basement membrane, and which sort of confirms the cell division again. That means the cancerous activities have started with the cells which are live cells. Although this is the statistics, these are several set of experiments. For example, if we look at experimental set 1, these are the live cells at the entry, and these are the live cells 48 live cells at the exit, 48%. And if through the same passage we do not allow the cells to pass, we keep them detained at the entry, then the natural death is indicated by this column, where we can see 86% live cells after the elapsed time. And that is where the cell viability was understood. And this is the, you can say, the pictorial view of what we did. This is the flow passage, the FEACM magnified view, and from the dye exclusion assay and dual staining, we could see that even though the cells are squeezed, deformed, but about 48 to 52% cells are live cells after crossing through these capillaries. That means they can start the activities even if 50% cells are live cells and all of them are not capable of starting cell division, but a significant number can start cell division. This is quite evident. Second part is basically through this process, when the cancer cells pass through different organs, there is a transformation that is called epithelial to mesenchymal transformation. Now, primary tumor cells are, from the primary tumor side, cells that come out are basically epithelial cells. This epithelial to mesenchymal transition is a process by which epithelial cells lose their cell polarity and cell-cell adhesion. Basically, it is added by proteins, and those proteins are sort of separated out and gain migratory and the invasive properties to become mesenchymal stem cells. And mesenchymal cells are deadly cells. We will show it little later. And this is this network of capillaries that is mimicked in this way. This is again on PDMS base. This complex channel and cavities were fabricated, and this time we could get real breast cancer cells. These are MD-AMB 468 cell lines. So you can see its growth from basically it is crossing one zone to another zone. It is crossing now this particular zone where we have some wells, and then heading towards this zone which are 35 micron diameter depth, and then finally it will pass through 7 micron network, and it will reach at the end of it. Now it is passing through this network and it will be collected at the outlet. So this is the total journey, and in this process we tried to understand EMT transition, epithelial to mesenchymal transition, and we used EFG for this, which is, you can see, epidermal growth factor. And what it does, it accelerates basically this transition. The epithelial cells are having sort of square or rectangular cross section, you can see, and we used about 20 nanogram per milliliter EGF, and finally you can see with 40 nanogram per milliliter and after 24 hours, these are all round cells, which indicates visually that these are mesenchymal cells, but we have to test it. And for that testing, what we did, this is bright field photography, and then we did some specific staining. This is calcine AM staining to stain the cytoplasm, and this is DAPI staining to stain the nucleus, and then merging these two images to get the merged image. And you can see the quite evident or quite convincingly you can understand the morphology of epithelial cells and EMT cells. EMT cells are smaller and round cells, and you can see the total cell here, how do they look really. Here again, while passing through, some of these cells are basically divided here itself into multiple cells, and and wherever clusters are there, clusters are broken, which confirm, or rather which indicates that basically mesenchymal cells are being produced. And then for further confirming it, we test it with two different protein bodies. One is fibronectin, another is vimentin. So EFG treated cells showed higher expression of vimentin and so these here we get, I have not mentioned it earlier, from the mesenchymal cells. Again we collect some mesenchymal cells, and those are passed through again microchannels, mimicking the passage through the bloodstream, collected at outlet and grown to study. So these are again, when mesenchymal cells are sent back through this capillary, there is a reverse transition. It gets, we get back epithelial cells. So this is EMT cells, these are MET cells, and these are epithelial cells. We can see that expression due to vimentin and fibronectin protein, these EMT cells or MET cells are almost same, whereas the epithelial cells, this black epithelial cells and MET cells are almost same, where we can see the expression for EMT cells. So we have written that also. Surprisingly, the vimentin and fibronectin levels reached the basal level as the epithelial cells, confirming MET transition. These are MET transition and these are EMT transition cells. So finally, why I made this comment that mesenchymal cells are extremely dangerous, because we can study this through colony formation assay, where epithelial cells and mesenchymal cells and also MET cells, both are all three samples are treated with the breast cancer medicine. It is basically one form of doxorubicin. It is called paclitaxel. So it is the medicine, so to say. So with paclitaxel, you can see epithelial cells, they are treated very well. Also you can see MET cells, they are treated very well, but response is quite poor with the mesenchymal cells. So it confirms the drug resistance behavior of MET transition. So this part, as I said, that can be summarized in this way, that through the complex network of capillaries, natural transition from epithelial to mesenchymal takes place, and it is understood that mesenchymal cells are drug resistant. and drug resistance is quite high, quite significant. So then in the third part, when this is treated with, basically we tried to test one commercial drug, paclitaxel, as you know, is the scientific name of the drug, but commercial drug is different names are there. What we get here is Paclicad, and this is a Paclicad flow of Paclicad particles, bright field image, and again basically microscopic image. And I'll come to the that part slowly. Slowly means gradually. So basically, castor oil was being used to make paclitaxel, but it is basically it's a resistance. So it reduces drug effectiveness. So we require to increase the dosage. Another option is to basically getting it bound through protein. And protein bound drugs, they act in both ways. The nonpolar region can bind the hydrophobic part, whereas the polar region can be favorable for the hydrophilic solvents. So this again the same test facility was used, and its movement through this microcapillary was observed, and some basic tests were done. As you can see through confocal microscope, basically these images were analyzed, and the particle size was found as 142 nanometer, whereas FESEM and FETEM the size was found as 132 and 138 nanometer, and these are also having charge. The zeta potential was measured. You can see average zeta potential varies from -5 microvolt to -43 microvolt. Here we can see a peak as a peak at -27 microvolt. And the only drawback I can show you, this is basically the protein bound Paclicad is passing through. You can see it passes through more smoothly, but at times these proteins get agglomerated and chokes the passage. So here you can see passage is choked. Also fortunately you will see, because one Paclicad particle will dissolve it and entire drug passes through. So this is one bottleneck to get it protein bound, that several capillaries can get choked, and clearing that part is little difficult. Here if you remember how single cells were passing through, that through convergent passage it was getting accelerated, it was getting decelerated. Same pattern is followed. Because of paucity of time, I will not go in much detail in that. And this was also, we did a fluid dynamic CFD study with basically agglomerate of Newtonian and FINIP fluids, and you can see the same with the actual path and what is obtainable through Newtonian fluid and what is obtainable to FINIP, which is viscoelastic fluid. We have plotted again, if you want to discuss, I can discuss. Time is less today. So we can quickly summarize this part also, that how to apply for the drug delivery. We can also, microfluidics is a very reliable tool, and here we have shown in a very succinct manner that how protein bound paclitaxel behaves, but in a more complex channel. Target is to design a more complex channel where with human blood this experiment can be done, and sort of organ on a chip kind of device can be developed.
Sir, I have question from part first. As you have mentioned, the transit zone capillary diameter around 7 micrometer and capillary length 200 micrometer. Then my question is, at what deformation index L by D the performance is optimum?
See, I mentioned about the capillary diameter.
Yes.
Capillary diameter, we used 7 micron. It ranges from 5 to 7 micron, and cancer cells, the average diameter, for example, breast cancer cells, MD, AMB 468 or 321, you can say average diameter 30 micron. Now, a 30 micron sphere, if it becomes a cylinder with 5 micron or 7 micron diameter, you can measure the elongation. We measured all these elongations. You can measure this deformation index. Yeah, that is how deformation is calculated.
Thank you, sir.
Here you can see the calculation of deformation index.
Hello. These cells are circulated along with blood.
Here it is DMEM medium.
Yeah, but in reality cells are circulated
Yes.
with blood,
Yes.
and then even the blood cells will squeeze through that one in the same way, because blood cell squeezing through this one was studied in other contexts, like Subra Suresh calculated how malaria drug.
Yes.
So when there are blood cells around these circulating cells, will there be agglomeration or interaction between them also, the cancer cell and the blood cell?
Usually not. Leukocytes at times found, because leukocytes are bigger than red blood cells.
RBCs,
Yeah.
red blood cells?
Red blood cells usually not.
So when we talk about this transition of M to E, epithelial to mesenchymal transition, so have we also have this discussion on, let's say, amount of deformation will affect this transition if we have more elongation, so as to say. Does it affect more? Means, do we have any discussion on or findings on how much deformation can yield how much of, how
Yeah,
many
very
cells?
nice question. See, basically, as I said, that when you get the epithelial cells from the primary cancer site or primary tumor site, then these are all bound by different kind of proteins, fibrogen, fibronectin, all these proteins, bind them. And if they pass through extremely narrow capillaries, then obviously these proteins get very quickly separated. And the agglomerate, when it's separated from protein and it becomes a single cell, it loses polarity, loses everything, and it becomes, more deadly for the human body. It becomes mesenchymal cells.
But the reverse case, sir, when we said when you pass it again, it reverses back to epithelial.
Reverse transition is still, a very unknown area, but through reverse transition, people who are specifically working on MET and MET signals or EMT signals, they found that, when these are again, collection of mesenchymal cells, again re-enter some such network, there is a possibility that they can, interact themselves. And which we also found without going into the detailed proof, because we, as I said, that here you can see that some collected cells, which were recycled, they have MET behavior, which are, equivalent to epithelial cell behavior, not like mesenchymal cells.
So, sir, in real body, let's say if we have pulmonary cell, arteries in series or something, do we have network of these pulmonary arteries where these cells will really squeeze in multiple times and then automatically the people can...
No, not through the same passage, but through another passage.
Yeah, I mean, if there's second passage...
After getting separated as mesenchymal cells, it may pass through another capillary, another network, where that can be converted into...
Does it happen in real body, does it happen in real case?
See, these are, any in vivo experiment is almost impossible. These are all in vitro experiments.
Okay.
Professor Sarit Das is here, a few other, people who work on cancer biology related areas. Some students are there, Shushmita, etc. I can see. Neeraj Prasad is another scientist who is working on cancer biology. So you can discuss with them. It is, in human body, it is conjectured. But all these experiments are done usually in vitro. Oh, Professor Sen is here. He can probably...
So I have a question. Thank you very much for the interesting talk. A mechanism for cancer spreading is that of cancer cells propagating through the extracellular matrix, which is like a porous matrix of collagen fibers. All right. So, and that has little to do with blood propagation. Right. So, and to my understanding, that is a major way of propagating, that is not through the blood capillaries, but through squeezing themselves through this sort of porous network of collagen fibers. So the question is, can you comment on this a little bit, that mechanism versus the mechanisms you presented today where the flow through capillaries?
No, surely. See, there is no doubt that through any conduit, if it is, vein or artery, from there to any other conduit, it is possible. We showed one extreme case, which is breast cancer, where the only possibility of reaching the cancer cells to the lungs from the surface of the body is through pulmonary capillaries. And that is why we, undertook that very specific special case. But as I said, in the other parts of the body, it can be any other conduit. As you rightly said, that obviously blood is a carrier. And from one organ to another organ, through blood vessels, if primary tumor site is such that it can release into a blood vessel, it will go through blood. But many a times it doesn't release through the blood vessels.
Yeah, thank you very much. Regarding the model and the validity of the model, a real blood vessel is an elastic object and you have a hard PDMS boundary. So I'm wondering, is a 5-micron blood vessel the same as a 5-micron PDMS channel, or should that be modeled in a slightly more elastic medium?
It is definitely, I admit, it is not, because blood vessels or capillaries, other pulmonary capillaries, they are flexible, lot more flexible.
So the effect should be less, right?
We just mimicked it with PDMS, and to bring about that change, little bit variation of that, amount of cross-linker was varied. You have more cross-linker, it is more rigid. If you reduce the cross-linkers, it is little more flexible. Only that part could be done. But, admittedly, this entire study is in vitro and absolutely preliminary study to understand. It can go to human level after, things are absolutely established and sure. But admittedly, blood vessels are basically, flexible, stretchable, and you have also pulsatile flow. We are not able to bring about those changes when we are trying to do this baseline experiment.
So if there are no more questions, so then let's thank Professor. Oh, there is one. There is one question. Yeah.
Thank you, Professor, for a nice presentation. So I have one quick question. You mentioned about the shape of these cells, right? Different shells. So when it becomes circular, it is more likely to go around or pass these conduits?
Yes, circular cells. See, this is very interesting. Many people ask me, this question, specifically this slide. These are, basically we redraw. This is the bright field picture of epithelial and mesenchymal cells. And calcin AM, this can stain only the cytoplasm. And DAPI stains the nucleus. When we merge, this is the merged picture. We can see the morphological difference between, epithelial cells and mesenchymal cells. Mesenchymal cells are round cells. We can very clearly see that after superimposing these two stained pictures.
So does it mean that once this transformation happen, it's something to the structural properties of the cell membrane, something like that happen?
Yes, yes.
So that can also affect the how it can deform.
Definitely. Definitely. And that, sort of, it is also at the same time cell divisions are there.
Okay. Thank you. Thank you, Professor Biswas. So, a small token of appreciation.
I will see in front of this audience, I have to admit something. I entered in this field. I do not know, whether I am known or unknown or whatever, but whatever is my entity, this biomicrofluidics, I entered in this field 5 years back. And primarily I got huge amount of help from two individuals. One is a professor in IIT Guwahati, Professor Siddhartha Shankar Ghosh, and second is again a very well known professor in IIT Kanpur, Professor Bushra Atik. So as such, entire work is done with the help of them, and one of the two of the students of us, me and Professor Bushra Atik, they are in the audience, and one student who is doing all the experiments with paclitaxel is Mr. Neeraj Prasad. He is also there in the audience. So this entire learning experience is through them. It is, but I presented them, not really I presented myself. Thank you.
Ladies and